So I was working with the L1000 data for the last couple of years, looking basically at some spatial patterns that I detected, and so I guess I can go right in. So nowadays that we measure gene expression on these multi-well plates, especially in the L1000 data, and I guess the bigger the data sets get, and with automation, we often see 96 well plates that have like 12 rows and 8 columns. And it's in the L1000 data, it's actually much bigger plates. It's 24 by 16 well plates. So it's great because we can scale up experiments a lot, but it comes with some issues that I want to highlight. Okay. So I guess everybody is familiar with the L1000 data and how it's measured with the Luminix speed assays. So I'm not going to talk too much about it. So in the following analysis, I looked at 1,890 plates from the most common cell lines that were in the first release of the L1000 data. So they're like on the bottom, these like 8 cell lines. And I looked specifically at the 3 types of data sets, which were knockdowns, drug perturbations, and overexpression studies. Okay, oops. All right, so the L1000 data processing pipeline, or like the different stages, they look something like this. So you have your raw data, and there is some deconvolution to kind of get the 1,000 genes from the 500 colors of the beads. And then after some normalization, we get this Q-norm data, which is basically just the landmark genes and their expression. So I will not talk about the inferred data at this point, but basically everything I talk about also applies to the data once it's inferred. Yeah. So we talk about the Q-norm data. So one thing is with biases, historically, they could be seen in other platforms as well. So with the Affymetrix chips, there's popular algorithms like the Mass5 or RMA to correct for these spatial artifacts. So here on the slide you can kind of see 2 examples of what can happen. So you can kind of see that the probe identity, the intensities can like vastly vary on the location on the chip. So in the first example, it's a thumb where somebody just touched the chip. On the other one, it's more like some machine error. So the good thing is with these arrays, they have a very high resolution in the X and Y direction. So that allowed for like a fairly easy correction of these probe intensities. But when we talk about the wells, like the plates here, the resolution is much lower, right? So here we only have resolution of 24 by 16, versus here where it's hundreds by hundreds, right? So I'm just gonna like really quickly show like how these biases look like on an individual gene basis, and then I'm gonna talk a little bit more about how we can look at the bias a little bit more different, and then talk about some algorithms that can remove that bias from the data. So this is 6 selected genes from 6 different plates in the L1000 data. And we can see that the gene expression is mostly a function of the location on the plate. So we have blue for like low expression and red for high expression. And I want to mention, so when I do this data analysis, I basically do a scale transform on the whole plate. So I'm using as control all the other samples on the plate. So I don't use the DMSO or weighted DMSO control. So yeah, so we can see that we get like a high spatial pattern, and it really isn't always the same. So every plate can exhibit a completely different shape of these biases. And then so if you see like a gray crossed out field, that just means that the data wasn't provided, or it probably didn't pass the quality control. So what we can already see is that the bias seems to have something to do with the quality control. And to this point, I haven't really figured out what can cause the bias, but I will talk about that later a little bit more too. So there's a big difference in the biases that were observed in the Affymetrix chip and in the bias that we have from the plates or the multiwell assays. So on these Affymetrix chips, for example, we have only two-dimensional data, if you want. So, but the two-dimensional data has a fairly high resolution. And on the other hand, with the L1000 data, we have a fairly low resolution for the plate rows and plate columns. But we have an additional dimension because we have multiple genes measured for each well. So the links data is really a three-dimensional bias problem versus the Affymetrix chip, which is just a two-dimensional problem. So we can kind of use the third dimension of this data to describe the bias better and more robust, and then remove it from the data. So as a quick concept, how do we quantify a bias on a plate? So the easiest way to quantify it is obviously to look at every individual gene of an experiment, and then for every experiment on the plate. And we can calculate something called the Moran's I autocorrelation. And so the autocorrelation is basically just a correlation of a variable to itself given a topology. So the topology in our case is the layout of the plate. So if I go back to the example of these genes, so if we pick any position on like any position, let's say for the first gene like TOP2A, if we pick a red one, we can see that all the values around it are also red. Or like if you pick a green one, most of the values will also be green. So the autocorrelation will basically give us something that's close to 1 in this case. So for the autocorrelation, values can range from -1, which would be kind of like a checkerboard pattern, to 1. So we can basically now go and like look at every gene on every plate on every sample and calculate the autocorrelation. So if we do that, we realize that like most of the plates in the L1000 will have some form of bias, and I will like go more into the detail once I go through all the required like formulas that we need to talk about some closer. So our main goal would be to increase the reproducibility of replicates in a batch. So the L1000 data usually runs in batches from 3 plates to 5 plates. And the plates are more or less identical, so it's the same drug perturbations, they're at the same location of the plate, and they were measured at the same time point, and they had the same cell line. So what I call the reproducibility score is basically the average correlation of all the replicates of the same perturbation across the samples of the plate. So each sample on every plate gets a reproducibility score. If there's any questions, you can ask in the middle. That's fine. So once we have this raw reproducibility score, which is since it's an average of correlation, it's something between 1 and -1. We can now correct it. So the reproducibility kind of depends on how much statistical power we have to actually detect true reproducibility. Depends also on the correlations to the other samples, right? So if a sample would have correlation of 1 to all the other samples, just like it's not meaningful that the replicates have also high correlation, right? So the replicate has to kind of stand out in its correlation from the background. So we can kind of compute like a random distribution of reproducibility scores for every sample across the batches. And we can do this by just sampling like random sets of samples and computing the reproducibility score. So we can build this distribution for every sample. So we can kind of see in the distribution here, we get something like a distribution that, you know, goes to like minus 0.3 to 0.3. And then if we would have a really good reproducibility for a sample, we would expect, you know, something that has a correlation that's higher than 0.2. Okay, so the blue curve is what we see before we do correction. And then once we correct, what we would expect is that we decrease the correlations, like random correlations of a sample to the other samples on the other plates. And that's generally what we will see. So we'll see a much narrower distribution. And then hopefully the replicates will maintain their correlation. And then if that's the case, we expect to get a higher reproducibility score after the correction. And then the delta reproducibility score basically is just the improvement that we get once we correct the gene expression. So X in our case is the original gene expression and X star is the corrected gene expression. Okay, so how would we want to approach this? So I mentioned before that the problem is technically a three-dimensional problem, which is nice. So so how can we describe the bias that we see, right? So we have this observed gene expression E hat, let's call it, and it's defined for a gene at a given well location, right? So on the plate it might be in the first row and the first column. And so the gene expression that we observe is basically the sum of the true gene expression, E, that we don't know, plus, and then we have this sum, which is basically a sum of bias functions, if you will, that will are additive to what we observe. So in this, so this bias is weighted for this gene G by a parameter C. And as you can see, this weight is constant for all the wells, right? So C is not dependent on I and J. But then we have this this class of functions that are basically functions over the location of the plate. Okay, so so when we have this, like when we accept it's kind of like a linear, like additive function, we can we can make this assumption, or we can hope that there's some redundancy in the biases, right? So this is kind of what this function describes, because here our like phi is not dependent on G, right? So like all of these functions affect every gene. It's just weighted for every gene separately, right? So these functions should be, you know, redundant for all the genes. So we can kind of assume, so this is exactly it, that like for C, it's usually positive. So it's never really zero. You know, like the absolute value can also obviously be a negative weight. Okay. So we go down, right. Okay, so what we can do is the following. So when we have this L1000 data, we basically have a matrix of 384 columns and about 1,000 rows for the landmark genes. So we can decompose this matrix X by just using singular value decomposition, and you know we get basically three matrices out of it: U, D, and V. And so the beauty about this decomposition is that it basically gives us exactly the definition that we had in the slide before. So I first talk about D. So D is obviously giving us the variance that's captured by the principal components, right? So we basically get the eigensamples times genes and the eigengenes by samples for U and V. But they actually directly translate into this matrix C and also to these functions. And I explain this real quick. So here the columns of V are the new components. It's the principal components of X. So what we now can do, instead of calculating the bias on the individual genes, the autocorrelation on the individual genes, we can calculate them on the principal component values. So because we do that, we have a much more robust measurement of the bias, and it allows us to get a much more accurate representation of the bias. So let's see. Yeah. So if we calculate the autocorrelation I for each column of V, given the topology of the plate, we can identify the components with a high bias, and those are the functions that we look for. So the key is to remove the biased functions or these biases from the data. So what we can do is we can set parts of the matrix D, which is the diagonal matrix with the variances, to 0 if we want to get rid of them. And then we can reconstitute the gene expression by just doing some matrix operations. So how we choose how much we remove from a bias, I will talk about in a bit. But yeah, so in the last slide, how you can see here, we reconstruct the gene expression by basically applying the weights to D, and then basically just back transforming the data. So it's a very simple approach. So how do we choose what we remove, right? So it depends a little bit on the data set. So the stringency of how quickly you remove a principal component based on its autocorrelation depends on the data. So I separated the knockdown, overexpression, and drug perturbation data sets, and I can basically maximize for getting the maximum average reproducibility score, if you want, after correction. So sigma is kind of telling us how stringent we are, how lenient we are with biased principal components. So in the first plot, you can kind of see that for the overexpression and drug perturbations in blue, we remove principal components once they, reach an autocorrelation of, 0.2, and then it starts to get more stringent. So if the principal component has a autocorrelation of 0.6, it gets completely removed from the corrected gene expression. For the knockdown data, we can actually be more stringent because it maximizes the reproducibility score, the mean reproducibility score for a more stringent parameter. So that's in red. So you can see that if a principal component would have an autocorrelation of 0.2, it nearly gets completely removed from the data. So on the bottom, you can kind of see how we identified the maximum. So if you look at the knockdown plot on the left, so at 1 we basically don't remove anything. So the gene expression basically stays the same, right? And then we slowly get more stringent with the lower the sigma gets, the more we shift our function to the left. So then we start seeing an improvement when sigma is 0.6. The reproducibility score improves. And then we reach the maximum at 0.15 for the knockdown data. And then after that, the reproducibility actually decreases. So we cut too much into the real signal, and we lose true biological meaning and reproducibility. So for the overexpression and the drug perturbations, sigma reaches its optimum at 0.35, and then the data degrades And like you can even degrade it more than the original data that you had. So you have to be careful how to choose that parameter. Okay. Oops. All right. So so what happens to the autocorrelation of the genes if we apply these biases? So this plot basically just shows yeah, what happens with like different sigma, and we can kind of see that if we don't do anything, the mean autocorrelation of genes is at 0.2, and so the more stringent we get, the lower the autocorrelation of the genes gets in the corrected gene expression. And then at blue we can see that, so this is the sigma that we use for overexpression and the drug perturbations. And we can't remove the bias completely. So the autocorrelation of the genes after correction is still slightly positive. But it's, it went from 0.2 to something that's more like 0.05 or 0.7. And the knockdown data, we could be much more stringent and still reach the maximum reproducibility. So here the autocorrelation of the genes could really be put to yeah, close to 0. Okay. So just want to show how this like works on artificial data. First, because this is like easy to visualize. So what we do is we generate a random matrix of 384 columns and 1,000 rows, and the data is just normally distributed. And then we, so this is our like true gene expression E that I had explained in the formula, and then we add these bias functions to the gene expression, which are functions over the location of the plate. So here we have 8 functions, and they're not orthogonal, so you know they won't build 8 principal components in the end, but they will build 4 principal components. So if we do our analysis and then we just plot the values of the principal components, those are the plots that we get on the bottom left. So not surprisingly, because we added these biases, there's bias in our gene expression. So if we do this randomly like 500 times, we can take the original gene expression and then correlate it to the modified gene expression after adding the biases. So this is what we see here in the bar plot, or in the box plot on the right. So our modified gene expression correlates to like 0.75 with the true gene expression. So after correcting the bias with the method that I explained, we can reconstitute the original gene expression nearly perfectly, right? And so the synthetic data is obviously easy because there is no correlations between genes to begin with, right? Random data, so these bias functions are really easy to isolate. But so this is kind of like the ideal case of what we try to achieve. So, okay, so how does it look like in the L1000 data, right? So if we look at all these plates, we looked at, I guess in this analysis, at 1,894 plates, and 96% of the plates had statistically significant bias. So, okay, so if we look at the plot on the left first, so this is the distribution of the autocorrelation of the genes in the 3 different data sets. So in red, we have the autocorrelation before correction, and then in yellow we have the autocorrelation after correction, and in gray we see what we would expect on random chunks. Okay, so we can see that especially in overexpression we have a very high average autocorrelation, and you know some genes have autocorrelation of 0.8, so they're like really biased on the location of the plate. Then after correction, as I said before, because we use a more stringent weight function on the knockdown data, the autocorrelation of the genes is is nearly random, like as we would expect in a random data set. And then for the drug perturbations and the overexpression, the genes show like a little bit of autocorrelation, but it is reduced. So the question is obviously, is this assumption of redundancy real, or do all the genes exhibit completely different biases? So what I show in the plot on the right is basically all the principal components ranked from highest variance to lowest variance captured, and it's only the first 100 principal components. And on the y-axis, I show the autocorrelation distribution of those first principal components for all the plates that we analyze. So, and then in the left column, it's the uncorrected data. In the right column, it's the corrected data. So what we see here is that actually the principal components that show the most bias on average are the high principal components. So the most biased principal component is the first component. And the autocorrelation there is 0.4 on average for all the 3 data sets that we looked at. So in general, like with gene expression, often the first principal component can carry something like 40 to 50% of the variance in the gene expression. And that is not true for the Lynx L1000 data. And this is, I think, mainly because the genes were selected to be independent from each other. So I think in total, the variance that has to be removed from the gene expression, looking at like the first, let's say, 10 principal components, is usually around 20 to 30% of the variance in the gene expression. That can be explained by bias. But that really depends on the plate. So, okay. So what happens if we correct? Can we improve the reproducibility? Right? So here I show the knockdown data, which has the most markedly positive effect from the correction. I will talk about the drug perturbations and overexpression in a bit. So here for the 8 major cell lines, you can kind of see on the x-axis is the reproducibility score before correction. And then on the y-axis we have the reproducibility score after correction. So if a plate improved, or like if a sample improves, so I do it on a single sample basis. If a sample improves, it should be above the diagonal. Right? So what we can see is that for samples that start off with a very low reproducibility score, they don't improve as much as samples that improve, that have already like a fairly high reproducibility score. They are improving even more. And so this is basically true for all the cell lines that we look at, and only some samples really decrease in reproducibility score. Okay, so the other 2 data sets are not quite as conclusive. And so this is something I kind of already started talking about. So I think in the drug perturbations, a lot of times the drugs don't really have any biological effect that's necessarily reproducible. So the correction will not really benefit them, right? So, when there was no signal before, after correction there won't be any signal either. So we don't really expect any improvement from samples that start off with a reproducibility score that is at 0, which would be random. So what we really have to look at are the samples that already have some residual reproducibility, and then ask if they improve. So for the overexpression, it's pretty easy to see that if they start off with a reproducibility of 5, let's say, they have a much higher reproducibility after correction. Yeah, for the drug perturbations, it's a bit hard to see in these plots, so I'll talk about that in like a little bit a different way. So, so how can we measure the success of an experiment? Like success, I mean the perturbation results in a measurable systematic change that is also captured by its replicates. Right? So in knockdowns and overexpression, there's a direct measure which we can look at, and that's just the gene expression of the target gene of the perturbation. So in the knockdowns, we would expect the target to be downregulated if the perturbation was successful. And in the overexpression, I would expect the gene expression of that target to go up. So for the knockdown data, that usually is pretty successful. So most of the genes show a significant downregulation. So here I only look at landmark genes. So if the target gene of a knockdown is a landmark gene, or of the overexpression is a landmark gene, I basically take the fold change of that gene and correlate it with the change in reproducibility score after correction. So what we get is exactly what we would expect. So we get a negative correlation of the fold change of the target gene with the increase of reproducibility score after correction. Because in a knockdown, the more a gene is downregulated, the stronger we would expect the reproducibility to be in the other experiments. And for the overexpression, we see positive correlation. It's even stronger. So the effect of the overexpression is pretty markedly, and it's because in the overexpression, a lot of the perturbations don't show any effect on the gene expression of the target. But when they change, when there's a successful overexpression, the experiments are very reproducible. And then also the correction has a much bigger impact. Okay, so for the drug perturbations, we don't really have a direct measure like this. So another way to look at this is in a little bit indirect way, and this is through the signature strength. And so for me, the signature strength, how I define it, is the sum of the absolute z-scores of a sample. So the more fold changes there are in a sample, the stronger the perturbation is. And this is actually a pretty good metric to identify if a sample improves from the correction or not. So as we can see here, it's like a heat map. So basically rank transformed the signature strengths of all the samples in the CPC data. And then it's basically a scatter plot with the change of reproducibility, also rank transformed. So you can kind of see that the samples that have high signature strength are also the samples that have the highest improvement in the reproducibility score. And this is also very nicely seen in the knockdown, even though there is this weird outlier. There's some that have a very high signature strength, but they don't really have any reproducibility improvement. So that's an interesting case there. So one of the issues and the differences between these three data sets is, if we look at the left plot, is that the similarity score or the reproducibility score that we observe is in the knockdowns is much higher than in the other two data sets. So this is why the samples of in a batch of a knockdown, they often correlate with 0.2, 0.3. But that's not necessarily the case for the drug perturbations and overexpression. So in the gray marked area, we have a majority of the samples of the overexpression and the drug perturbations. So if I go to the right, we can kind of ask, if we bin the samples based on their initial reproducibility score, and then ask how much they improved from reproducibility, we kind of see that samples that have a high reproducibility score generally improve more. And we can actually see this in the overexpression very nicely, that even though there's very few samples that start off with initial a high reproducibility score, they really improve quite a lot, nearly as much as the knockdown. The knockdowns just generally perform better even in the samples that don't have very high reproducibility score to begin with. So we couldn't really figure out why that was. So okay, so the question is this really that relevant to downstream analysis? And so it's actually if you do enrichment analysis on the signatures that you derive from this z-score transformation, the enrichment algorithms like gene set enrichment are actually quite sensitive to these biases. So if we look at this, so this is just taking 2 example plates. So these are just randomly selected. And then there is 2 biological processes that we look at. So if there is a plus, it's a significant upregulation. And if there's a minus, it's a significant downregulation of the biological process. So what we can see is that these biases actually correspond to biological, meaningful entities, which is problematic, because if it would be, random genes that don't really have anything to do with each other, these statistical methods wouldn't necessarily pick up on them. But in this case, we can see that enrichment is very sensitive to it. So say the bottom left panel, you can kind of see that all the positively enriched terms are on the right and all the negative on the left. But in this case, the drug perturbations are completely randomized on the plate, so we wouldn't expect this pattern. And then after the correction, first of all, we have much fewer enriched signatures, and the signatures intermix much more. In the other case, on the top, actually we don't have any enriched signatures after correction. And so as we've seen, so we can also apply the autocorrelation on the enriched terms. So if we calculate the enrichment score for every sample on the plate, we can just calculate the autocorrelation. And so that's the plot on the top right. So you can kind of see it's very similar to the individual gene bias. There is a bias in the enrichment scores in the uncorrected data. And that autocorrelation is removed significantly after the gene expression correction. Okay, so what are frequently biased processes? So I want to mention first that we were a little bit limited in what we could calculate in terms of enrichment, because we only had the L1000 genes. So if you do gene set enrichment analysis or you do Fisher exact test, it doesn't really matter. Since we only look at 5% of the genes, we can only work with biological processes that had sufficiently large numbers of genes to allow a statistically significant overlap. But so we see a lot of homeostasis. So homeostasis, for example, has an average autocorrelation in the enrichment over all the plates of 0.28. So it's quite significant, I would say. So I think so from this slide, I think the main take-home message is that the bias is caused by something like cell viability. So it's not the technology. It's not something that stems from the Luminix bead assay or something, or the number of beads that were extracted. It's really growth conditions on the plates, and the cells are responding often stronger to these effects than to the perturbations themselves. So the most of the signal that we see in the gene expression can really be explained by the location of the plate. So so here we try to delve a little bit more into the meaning of these principal components. We try to kind of identify if we could identify what causes the bias. Is it temperature or light? So it suggests that it's some kind of gradient, because that's usually what we see on these plates. So it should be something like temperature or humidity maybe. So in this plot, what we did is we extracted from the 1,890 plates, we extracted all the principal components that we would remove by at least 50%. So they would have to have some significant bias in them. And so we ended up with about 2,500 principal components that showed significant bias. And so they came from all the 8 different cell lines. And so this matrix that you see has 2,500 columns. And then we just plot highly biased or highly associated enriched biological processes to them. So one thing that we can see is that the principal components that we find in all these different plates seem to be related to each other. So like on the left, there's this group of principal components that seem to be enriched in the same biological processes. And if you would go and calculate the correlation, or if you would calculate the correlation of the genes of the gene coefficients, they would be highly correlated. So the principal components that we find are conserved across the different plates. So yeah, so in the sense, so at this point we don't really know like what the bias is, but we know it has to be something that kind of shows like a response of the living cells on the plates and kind of reflects the different microenvironments on a plate. Even though it should be like a very controlled environment, there's enough variability to add like a significant amount of variance to the gene expression. So I have a small example. So we have a small data set of 6 plates. And I'm not going to talk too much about like what it is, but so usually when you have the L1000 data, you get these quality checks, and like in terms of the technology, all the quality controls pass pretty much, and we can see like if we look at median count per well, there's no apparent problem in like the counts, or like the invariant set of the 10 levels like looks fine. So we don't really see any of these patterns showing up in the quality control. But so if we map the principal components onto these plates, so here I'm showing the first principal components of the plate, and we can see clear biases that have to be removed from the gene expression. So for this plate, for example, if I rank the principal components by their variance from 1 to 400 or like 384, and then plot their autocorrelation, we can see that the first 10 or so principal components show like a clear increase in autocorrelation. And then after that, the autocorrelation of the principal components is actually fairly low. It's like median around 0. So this is already what we saw in these other plots, that the first principal components are usually the most biased. And so in our data set, this is more or less true for all the plates that we have. And so if I run a correction, so if I run this correction, for this data set I would remove 27% of the variance from the gene expression. So again, it's like it's something you wouldn't expect to be only 26%, because if you look at it's like the first 10 principal components that are biased. But then again, that's like a property of the L1000 data, that the principal component variances are pretty flat. So I think the first principal component captures 8% of the variance or something. So that's it's an unusual distribution for variances of principal components. Which means it's a good thing, right? So you don't lose too much information when you remove the bias from the gene expression. Just for good measure, so these are 2 plates from the same batch, and you can kind of see that the bias always looks a little bit different, right? So you can't predict, that the bias will always go from the left to the right, or like from the bottom to the top. Sometimes it starts in the center and it grows out radially. Sometimes, as you can see on the bottom right, you have these edge effects, right? So this is like the edge effect is kind of like a known problem, where the humidity on the outer wells is not the same as inside the plate. So often in experiments, people will not even plate those. They will just put water in it. It's just 2 different. It's basically the same plate design. It's the same cell line, same time point, but the biases are distributed completely different. So one of the issues, I'm nearly done now actually, but so one of the things I want to mention, and this is like about the plate design of L1000, or like any plate design. If you design any experiments like this, is that you have to be careful how you place samples on these plates. So if you want to use this method and you want to use autocorrelation, you shouldn't put replicates next to each other on the plate. So usually, experimentally, I think for experimentalists, it's a convenient thing to do. So I've seen a lot of plate designs where, the replicates are right next to each other in like neighboring wells. And like when you see data like this, you would really think you should really randomize the positions of your drugs. Ideally, you run an algorithm that gives you something randomized. And then also, so in our data, for example, the titrations of the same drugs are next to each other. So that can kind of bias this analysis because let's say you have 5 different concentrations of the same drug always next to each other. The autocorrelation can be skewed and it can overestimate the bias that you see. But like in our case, like when you have something like on the upper left with a gradient, that's most likely not from similarities of the perturbations that are next to each other, but really like a true bias. But you just don't know. And like so this is on, so this is kind of a luxury to have so much data to analyze, and having these big plates helps a lot. So 24 by 16 is working much better for detecting biases than if you only have a 96 well plate. So on the 96 well plate, what happens is that your random reproducibility score is much wider, right? So this really helps. Like having all these samples on the same plate really helps to statistically identify the bias. Yeah, so that's about it. So if you have questions, let me know.
Thanks, Alexander. That was excellent. Questions, anybody? Well, I have a question. Oh, I'm sorry. Go ahead.
Thank you so much, Mario. This is Jane Bai from FD. Very nice presentation. I'm just wondering, you did a lot of computations, corrections. Will you publish corrected gene expressions somewhere with all those autocorrelations available for people to use?
Oh, I see. Okay, so I did not. So because the data is so large, I did not publish any corrected data sets. So this is a published paper. So if you go to, I have it on the shot. So it's like right here. It's in Bioinformatics. It was published like 2 months ago or 3 months ago. And it basically has like this presentation. It has like a fairly detailed supplementary material section, and it has R scripts. It should be pretty easy to run. So if you if you're interested in like special data sets, you can just run the correction really quickly. So the whole, like correcting the whole data set would probably take 4 hours, or maybe like a day on a computer. So it's not very computationally intensive. But I mean, if there's interest, we could probably publish a whole corrected data set. What I could not provide is I could not correct the data and then infer the landmark genes because we don't have the model. Infer like the rest of the genes because we don't have the model.
Thank you.
No problem.
So that took care of my first question.
Okay.
The second question was, so you talk about this background distribution that you generate by randomly looking at random pairs of perturbations, but you don't seem to use it actually in your...
Right, so it's, let me show, okay, so here, so this RS star, right, the reproducibility score, if it's uncorrected, is a value of -1 to 1, right? And so like all the other times I showed the reproducibility score, it was the corrected reproducibility score. So then the values are ranging from, like something negative to something positive. So in this case, they go to like 25. So usually I use the normalized reproducibility score, because the reproducibility score uncorrected doesn't really mean much, because you have to take into account how significantly the similarity outlies the background distribution.
Right, but I guess I didn't see where you explicitly do this normalization, or that's implied somewhere.
So this is so the reproducibility score I really only use as a benchmark.
Okay.
And to infer also the optimal sigma, right? So for example, here if I try to identify sigma, I calculate the change in the mean. Okay, sorry, it doesn't say, but it's the mean normalized reproducibility score. So finding a maximum in just the reproducibility score wouldn't work, because you actually remove correlations from the data. If you remove variance, you generally remove some variance. So if you look at the absolute correlation of the replicates, you might actually reduce the correlation a little bit. But the important thing is that you remove much more correlations from samples that are not true replicates.
Right. No, yeah, that makes perfect sense. I just never, I heard you say that you're actually...
Yeah, I can. Yeah.
Yeah. Thanks.
Can you hear me? I have a question.
Yeah, I can hear.
Mark Archer, this is certainly a cautionary tale, and I wonder if you can use this method to define genes that you believe. You certainly show us a lot of things that are not believable. Can you isolate those that are and that we can trust?
So in general, so most of the things I've shown are the principal component values, right? Which principal component is is all the genes together. So sure, I mean, like because like if we look at this, right, we kind of identify that there's certain principal components that that seem to be conserved over like all the plates, right? So obviously then I can, I could rank the genes by likelihood of being biased. The question is, so this is something that might like be worth like looking more into. If a gene that's likely to be biased is untrustworthy after correction, I don't know. I don't know if you have to dismiss. I don't know if you have to dismiss like 30% of the genes because they contribute to a bias frequently.
And there's a lot of cellular processes in there that would be expected to be interesting or affected by drug perturbations.
I mean, so this is also a problem of the method, I think, right now. I mean, if I can probably find an example. Yeah, so like if we look at the first principal component of this plot, you see the blue one, right? Like it's like this blue dot. So this principal component is marked for removal, right? The autocorrelation is so high that basically all the information of this is deleted. But for this sample here in blue, there might be a true effect that is just the same as the bias, but we lose that information in the correction, right? So maybe there's like ways we could be more careful about removing or like handling these cases. Right on the bottom right, you see the same where there's this like blue sample, right?
Right, and they're isolated. They're not surrounded by other blue squares.
Exactly. But so in this case, it would just be, so that effect would be removed. So if this is a very characteristic effect of that sample, it's lost. But this is like the first, it's like the paper was mainly there to kind of make people aware of the bias. And so just want to clarify, so this is not only L1000, so this is any experiment that's run on plates. So I've seen plates with RNA-seq where you also see biases. So it's not, this is not exclusive to the L1000. This is just the only data set that allows like a systematic analysis of that effect.
It's a nice study. Thank you very much.
I just want to make two comments. One, and I have a question, and So one comment is like this data is like a brand new data that we submitted from here to processing with the L1000 from 2 different labs at Mount Sinai. So this is sort of like our own data that we are going to analyze soon. The other comment is like Alex said something like we don't have the function to infer the rest of the genome. So from what I understand, Mario, we have it, right? Like we have the full pipeline, including the inference model in as an open source pipeline. Is that something that is correct?
Yeah, that's correct. We do have the pipeline. The question is, and I kind of didn't think about it, would the inference model that was derived for uncorrected scores work the same? Or you would have to come up with that model, right?
So I think it would be the same because the model is independent of the bias, but it was inferred from like 100,000 different gene expression studies. So I would assume that the correlation structure of the genes stays the same. I mean, that should be fine.
Yeah, then it would be rather straightforward. Yeah, there are MATLAB on the Broad's GitHub.
Yeah. So I know. So the thing is, I know that the function is in the library, but the model itself is a data set, right? It's a full linear model, and I don't think that file. I never saw that file. I only saw the MATLAB script to infer the gene expression, but I never saw the model itself. I'm not sure if that's in the repository.
Okay, but that's all you need, right? You need the script that uses the model, right? So,
right. Yeah, but this is the thing. I don't have the coefficient linear model.
It's all there. So you know.
Okay. Good.
Yeah, we tested the pipeline. So they have a relatively new pipeline that was deposited maybe 6 months ago, and right, you can do it all.
Okay. I mean, one of the things is, I mean, as somebody was mentioning, like if it's a good idea to maybe identify genes that are commonly biased and replace them with genes that are not biased as frequently. I mean, that could be maybe a possible modification of the protocol.
Right, but what it looks like is that bias, and I think you're making a good point. It's biological processes that are shifted, and it looks like, you know, these cell growth processes are affected, which could be that basically in parts of the plate cells live better than in other parts of the plate, and then, you know, you can't really fix that by removing genes, I think. I don't know.
Yeah.
Okay, well, we're just on time. Thank you so much, Alexander. That was brilliant.
Thanks a lot.
All right. We'll get this webinar on the website as soon as we can. So if you guys want to look at the details, which I will definitely want to see it again, so it will be available. Thanks.
All right.
Thanks.
Thank you.
Thank you.